everyone. Welcome to a new session of the paper Cell Biology. In this module, we would study how the cells interact with each other, the various methods by which cells move with special reference to muscle and nerve cells. We would also study about the process of cell death and cell senescence. The process of cell differentiation in plants and animals would also be studied. Cell to cell interactions. To make a multicellular organism, cells must communicate just as humans communicate if they are to organize themselves into a complex society. And just as human communication involves more than the passage of noises from mouth to ear, cell-cell communication involves more than the transmission of chemical signals across the space between one cell and another. Complex intracellular mechanisms are needed to control which signals are emitted at what time and to enable the signal receiving cell to interpret those signals and use them to guide its behavior. In this module, we would look in detail at how cells of multicellular organisms interact with each other and also examine the ways in which cell surfaces interact to organize tissues and body structures. Cell signaling with chemicals. Communication between cells is common in nature. Cell signaling occurs in all multicellular organisms, providing an indispensable mechanism for cells to influence one another. The cells of a multicellular organism use a variety of signals, including not only peptides but also large proteins, individual amino acids, nucleotides, steroids and other lipids. Cell surface receptors. Any given cell of a multicellular organism is exposed to a constant stream of signals. At any time, hundreds of different chemical signals may be in the environment surrounding the cell. However, each cell responds only to certain signals and ignores the rest. Located on or within the cell are receptor proteins, each with a three-dimensional shape that fits the shape of a specific signal molecule. When a signal molecule approaches a receptor protein's shape, ultimately producing a response in the cell. Hence, a given cell responds to the signal molecules that fit the particular set of receptor proteins it possesses and ignores those for which it lacks receptors. Types of cell signaling. Cells communicate through any of the four basic mechanisms: direct contact, paracrine signaling, endocrine signaling and synaptic signaling. Direct contact. When cells are very close to one another, some of the molecules on the cell's plasma membranes may bind together in specific ways. Many of the important interactions between cells in early development occur by means of direct contact between cell surfaces. Paracrine signaling. Signaling molecules released by cells can diffuse through the extracellular fluid to other cells. If those molecules are taken up by neighboring cells, destroyed by extracellular enzymes or quickly removed from the extracellular fluid in some other way, their influence is restricted to cells in the immediate vicinity of the releasing cell. Signals with such short-lived local effects are called paracrine signals. Like direct contact, paracrine signaling plays an important role in early development, coordinating the activities of clusters of neighboring cells. Endocrine signaling. If a released signal molecule remains in the extracellular fluid, it may enter the organism's circulatory system and travel widely throughout the body. These longer lived signal molecules, which may affect cells very distant from the releasing cell, are called hormones, and this type of intercellular communication is known as endocrine signaling. Synaptic signaling. In animals, the cells of the nervous system provide rapid communication with distant cells. Their signal molecules, called neurotransmitters, do not travel to the distant cells through the circulatory system like hormones do. The narrow gap between the two cells is called a chemical synapse. While paracrine signals move through the fluid between cells, neurotransmitters cross the synapse and persist only. Intracellular receptors. All cell signaling pathways share certain common elements, including a chemical signal that passes from one cell to another and a receptor that receives the signal in or on the target cell. Many cell signals are lipid soluble or very small molecules that can readily pass across the plasma membrane of the target cell and into the cell, where they interact with the receptor. Some bind to protein receptors located in the cytoplasm, others pass across the nuclear membrane as well as bind to receptors within the nucleus. These intracellular receptors may trigger a variety of responses in the cell depending on the function of the receptor. Receptors that act as gene regulators. Some intracellular receptors act as regulators of gene transcription. Among them are receptors for steroid hormones such as cortisol, estrogen and progesterone, as well as the receptors for a number of other small lipid soluble signal molecules such as vitamin D and thyroid hormone. Because of their structural similarities, they are all part of the intracellular receptor superfamily. Each of these receptors has a binding site for DNA. In its active site, the receptor typically cannot bind DNA because an inhibitor protein occupies the binding site. When the signal molecule binds to another site on the receptor, the inhibitor is released and the DNA binding site is exposed. The receptor then binds to a specific nucleotide sequence on the DNA which activates a particular gene, usually located adjacent to the regulatory site. The lipid soluble signal molecules that intracellular receptors recognize tend to persist in the blood far longer than water soluble signals. Most water soluble hormones break down within minutes and neurotransmitters within seconds or even milliseconds. A steroid hormone like cortisol or estrogen, on the other hand, persists for hours. Receptors that act as enzymes. Some intracellular receptors act as enzymes. A very interesting example is the receptor for the signal molecule nitric oxide. Nitric oxide diffuses readily out of the cells where it is produced and passes directly into neighboring cells where it binds to the enzyme guanylyl cyclase. Binding of nitric oxide activates the enzyme, enabling it to catalyze the synthesis of cyclic guanosine monophosphate or GMP, an intracellular messenger molecule that produces cell specific responses such as the relaxation of smooth muscle cells. Cell surface receptors. Most signal molecules are water soluble, including neurotransmitters, peptide hormones and the many proteins that multicellular organisms employ as growth factors during development. Water soluble signals cannot diffuse through the cell membranes. Therefore, to trigger responses in cells, they must bind to receptor proteins on the surface of the cell. These cell receptors convert the extracellular signal to an intracellular one, responding to the binding of the signal molecule by producing a change within the cell's cytoplasm. Chemically gated ion channels. Chemically gated ion channels are receptor proteins that ions pass through. The receptor proteins that bind many neurotransmitters have the same basic structure. Each is a multi-pass transmembrane protein that threads back and forth across the plasma membrane. In the center of the protein is a pore that connects the extracellular fluid with the cytoplasm. The pore is big enough for ions to pass through, so the protein functions as an ion channel. Enzyme receptors. Many cell surface receptors either act as enzymes or are directly linked to enzymes. When a signal molecule binds to the receptor, it activates the enzyme. In almost all the cases, these enzymes are protein kinases, enzymes that add phosphate groups to proteins. Enzyme receptors have a common structure, that is, they possess a single transmembrane protein. The portion that binds the signal molecule lies outside the cell, and the portion carries out the enzyme activity is exposed to the cytoplasm. G protein linked receptors. G protein coupled receptors or GPCRs, also known as 7 transmembrane domain receptors, 7 TM receptors, heptahelical receptors, serpentine receptor and G protein linked receptor or GPLR constitute a large protein family of receptors that sense molecules outside the cell and activate inside signal transduction pathways and ultimately cellular responses. They are called 7 transmembrane receptors because they pass through the cell membrane 7 times. Interaction of cells with other cells. Examination of a thin section through a major organ of an animal reveals a complex architecture involving a variety of different cells. Very little information is available regarding the mechanism responsible for generating the complex three-dimensional cellular arrangements found within the developing organs. Dozens of different proteins involved in cell adhesion have been identified. 4 distinct families of integral membrane proteins play a major role in mediating cell-cell adhesion. 1. Selectins. 2. Certain members of Immunoglobulin Superfamily or IGSF. 3. Certain members of the integrin family. 4. Cadherins. Selectins. Selectins comprise a family of integral membrane glycoproteins that recognize and bind to a particular arrangement of sugars in the oligosaccharides that project from the surfaces of other cells. Selectins possess a small cytoplasmic domain, a single membrane spanning domain and a large extracellular segment that consists of a number of separate modules. There are 3 known selectins: E-selectin present on endothelial cells, P-selectin present on platelets and endothelial cells, and L-selectin present on leukocytes. Binding of selectins to their carbohydrate ligands requires calcium. Selectins mediate transient interactions between circulating leukocytes and vessel walls at sites of inflammation and clotting. The Immunoglobulin Superfamily. Antibodies, which are a type of protein called an immunoglobulin, were found to consist of a polypeptide chain composed of a number of similar domains. Each of these immunoglobulin domains, as they are called, is composed of 70 to 110 amino acids organized into a tightly folded structure. The human genome encodes 765 distinct immunoglobulin domains, making it the most abundant domain in human proteins. Taken as a group, these proteins are members of the Immunoglobulin Superfamily or IGSF. Most members of Immunoglobulin Superfamily are involved in immune function, but some of these proteins mediate calcium independent cell-cell adhesion. Most of the Immunoglobulin Superfamily cell adhesion molecules mediate the specific interactions of lymphocytes with cells required for an immune response. However, some IGSF members such as VCAM or vascular cell adhesion molecule, NCAM or neural cell adhesion molecule and L1 play important roles in nerve outgrowth, synapse formation and other events during the development of the nervous system. Cadherins. The cadherins are a large family of glycoproteins that mediate calcium dependent cell-cell adhesion and transmit signals from the extracellular matrix to the cytoplasm. Cadherins typically join cells of one type to another and do so predominantly by binding to the same cadherin present on the surface of the neighboring cell. Like selectants and immunoglobulin superfamily molecules, cadherins have a modular construction. The best studied cadherins are E-cadherin or epithelial cadherin, N-cadherin, neural cadherin, and P-cadherin or placental cadherin. Structural studies indicate that cadherins from the same cell surface associate laterally to form parallel dimers. Different types of cells bearing different cadherins likely engage in different types of interactions. Cadherin mediated adhesion is thought to be responsible for the ability of like cells to sort out of mixed aggregates. Cadherins may be the single most important factor in molding cells into cohesive tissues in the embryo and holding them together in the adult. Adherin junctions and desmosomes. The cells of certain tissues, particularly epithelia and cardiac muscle, are very difficult to separate from one another because they are held together tightly by specialized calcium dependent adhesive junctions. There are two types of cell-cell adhesive junctions, adherens junctions and desmosomes. Adherin junctions are found in a variety of sites within the body. In adherens junction, cells are held together by calcium dependent linkages formed between the extracellular domains of cadherin molecules that bridge the 30 nanometer gap between neighboring cells. The cadherin clusters of an adherens junction connect the external environment to the actin cytoskeleton and provide a pathway for signals to be transmitted from the cell's exterior to the cytoplasm. Desmosomes. Desmosomes are disc shaped adhesive junctions approximately 1 micrometer in diameter that are found in a variety of tissues. Desmosomes are particularly numerous in tissues that are subjected to mechanical stress such as cardiac muscle and the epithelial layers of the skin and uterine cervix. Tight junctions. Tight junctions are specialized contacts occurring between neighboring epithelial cells. Tight junctions are located at the very apical end of the junctional complex between adjacent epithelial cells. Tight junctions serve as a barrier to the free diffusion of water and solutes from the extracellular compartment on one side of an epithelial sheet to that on the other. Tight junctions also serve as fences that help maintain the polarized character of epithelial cells. They are also present between the endothelial cells that line the walls of capillaries. These junctions are particularly evident in the brain where they help form the blood-brain barrier. Cells of the immune system are able to pass across the endothelium through these junctions. Plasmodesmata. Unlike animal cells, plant cells are separated from one another by a substantial barrier, the cell wall. Even though plants lack the specialized junctions found in animal tissues, most plant cells are connected to one another by plasmodesmata. Plasmodesmata are cytoplasmic channels that pass through the cell walls of adjacent cells. Like gap junctions between animal cells, plasmodesmata serve as sites of cell-to-cell communication as substances pass through the annulus surrounding the desmotubule. Let us now summarize what we have learnt in this session. Direct interactions between cells as well as between cells and the extracellular matrix are critical to the development and function of multicellular organisms. Some cell-cell interactions are transient, such as the interactions between cells of the immune system and the interactions that direct white blood cells to the sites of tissue inflammation. The binding of a signal molecule to an intracellular receptor usually initiates transcription of specific regions of DNA, ultimately resulting in the production of specific proteins. Here are a few questions for you to work out. 1. Explain the different types of cell signaling. 2. Explain as to how the receptors can act as gene regulators. 3. Compare the arrangement of integral membrane proteins in a tight junction versus a gap junction. 4. Explain the role of Plasmodesmata. 5. Describe the different types of selectants and its role in cell-to-cell interaction. Here are a few books and websites for your reference. The Cell, a Molecular Approach, 3rd edition, written by Jeffrey M. Cooper, Robert E. Hossman, ASM Press. Biology, 5th edition, written by Peter H. Raven, George B. Johnson, published by McGraw-Hill. Cell Biology, 7th edition, written by Gerald Karp, published by John Wiley and Sons. Here are a few websites for your reference. www.senescence.info/cellaging.html, www.embryo.asu.edu/pages/embryonic differentiation-animals, www.biologyreference.com. Thank you for watching this program. Let's meet in the next session with another topic. Till then, bye.